Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a really key malaria drug, one known for lasting a long time to protect people, but maybe it's starting to lose its power over time.
Uh-huh. That's a worrying thought, especially with a drug like piperaquine.
Exactly, piperaquine. It's been so important because it, hangs around in your system, giving that longer protection against getting infected again.
Yeah, that long-lasting action is precisely why it's become such a vital tool, especially in places where malaria is, well, constantly a threat.
Right. But here's the twist. Some new research suggests that very strength, its long duration, might actually become a weakness under certain conditions. So for this deep dive, we're looking closely at a study out of the Karolinska Institutet, published in Nature Communications. We've got a health news summary and, crucially, the actual scientific paper itself.
And our aim today is pretty clear, isn't it? We want to unpack what these scientists found out.
Yeah, what did they discover?
We need to explore how using dihydroartemisinin and piperaquine again and again might actually reduce how well it shields you from getting malaria later. And also, what does this mean for fighting malaria overall?
Okay, let's dive in. What was the study really trying to get at? What was the core question?
So the main thing was, does treating someone multiple times with dihydroartemisinin and piperaquine change how well the piperaquine part prevents future infections?
Okay.
And importantly, do changes in the malaria parasite's genes, its genetics, have anything to do with that potential drop in protection?
Right. So how did they try to answer that? What was the method?
They did a retrospective analysis, basically looking back at data that was already collected.
Okay, from where?
From a big clinical trial called WANCAM. It happened in southern Mali between 2012 and 2014.
Right.
And in that trial, people who kept getting malaria were treated with either dihydroartemisinin and piperaquine or another combination drug, artesunate amodioquine.
So they had these two groups already from the old trial data. What did they compare?
They compared what happened to the patients in the dihydroartemisinin and piperaquine group versus the artesunate amodioquine group. They looked at how often people got infected again.
Reinfection rates.
Exactly, reinfection rates, and also how long it took for that reinfection to happen. They tracked this actively with checkups and passively, like when people felt sick and came in.
And you mentioned parasite genetics.
Yes, that's key. They did molecular analysis on the Plasmodium falciparum parasites from these infections.
Molecular analysis, meaning they looked really closely at the parasite's DNA.
Pretty much. They were specifically hunting for changes in the number of copies of certain genes, plasmepsin 2 and plasmepsin 3.
Plasmepsin 2 and 3. Why those genes?
Because they're thought to be involved in how the parasite deals with piperaquine. So by looking at these genes over time in relation to the treatments, they hope to figure out why the drug's protective shield might be weakening.
Okay, so they compared the two drugs, tracked reinfections over 2 years, and looked at changes in these specific parasite genes. Makes sense. What did they find? What were the big takeaways?
Well, a few really important things emerged. First, at the start, dihydroartemisinin and piperaquine did give better protection against getting reinfected compared to the other drug.
Which you'd expect because of the piperaquine lasting longer, right?
Exactly, but, and this is the crucial part, that advantage didn't hold up over the whole 2 years. The protection from dihydroartemisinin and piperaquine actually seemed to fade over time.
It faded. How did they measure that?
Well, they saw that the risk, the likelihood of getting malaria again in the group, getting dihydroartemisinin and piperaquine, went up compared to the other group as the study went on.
Okay.
And maybe even more telling, the average time it took for someone in that dihydroartemisinin and piperaquine group to get reinfected got significantly shorter between 2012 and 2014.
But people were getting sick again faster.
Yes, faster than they were at the beginning of the study period.
Wow. Okay, that definitely sounds like a decline. And did they connect this back to those plasmepsin genes?
They did. It seems this decrease in protection happened alongside an increase in infections caused by parasites that had extra copies of the plasmepsin 3 gene.
More copies of plasmepsin 3.
They found these parasites, the ones with extra copies, were more common in the reinfections that happened relatively soon after someone was treated with dihydroartemisinin and piperaquine.
Interesting.
And they also saw more variation, more changes in the copy number of plasmepsin 2 in repeat infections in that same drug group, especially when the time between malaria episodes was short.
So it really looks like the parasites might be, finding ways to cope when they keep bumping into this drug combination.
That's certainly what it suggests. It's like the drug pressure is selecting for parasites with these genetic changes.
Did they find anything else genetically? Like things seen elsewhere?
Well, interestingly, they didn't find that specific plasmepsin 1,3 hybrid gene structure that's been linked to piperaquine resistance over in Southeast Asia. That wasn't present here.
Yeah.
And they looked at another gene, PSCAT, where certain mutations sometimes correlate with faster reinfection. But it didn't look like piperaquine was strongly pushing for those particular mutations during this specific study in Mali.
Got it. But one thing you mentioned earlier, and it seems really important, is that even while this preventative effect was maybe weakening,
Yes.
the drug combination, dihydroartemisinin and piperaquine, was still actually working well to clear the initial infection, right?
Absolutely crucial point. The study confirmed that the drug's ability to treat the malaria you have right now remained high throughout the trial. It was still effective therapeutically.
So it cures the current illness.
Yes. The worry is specifically about its declining power to prevent the next infection, especially when it's used over and over again.
Okay. So that leads us to the bigger picture. What are the implications here? Why should we be paying attention to this?
Well, the researchers raise a really big flag about that long duration of piperaquine in the body. What was seen as a huge plus for prevention
Right, the long half-life.
might, paradoxically, be helping parasites develop tolerance when the drug combination is used repeatedly in places with lots of malaria transmission.
How would that work? Tolerance, meaning not quite resistance, but getting tougher.
Kind of. The idea is this. The dihydroartemisinin part clears out relatively quickly, but the piperaquine lingers for much longer.
Okay.
So there's this period where parasites are exposed only to piperaquine, maybe at levels that aren't high enough to kill them all outright, especially if they have some sort of advantage.
Ah, like those extra copies of the plasmepsin 3 gene.
Exactly. That lingering lower-level piperaquine creates an environment, a sort of selection pressure, where parasites with those protective gene duplications are more likely to survive and eventually cause a new infection. Repeat this cycle, and those tolerant parasites can become more common.
Wow, okay. It's like evolution happening right there, driven by the drug itself. So what does this mean for big public health programs that rely on that long-lasting effect?
That's the major concern. Think about strategies like intermittent preventive treatment for pregnant women or seasonal malaria chemoprevention for kids. These often use piperaquine specifically because it lasts long, giving repeated protective doses.
Yeah, they're designed around that feature.
Right, but this study suggests we need to be really cautious. Using piperaquine this way repeatedly across large populations could strongly select for those plasmepsin 3 duplications.
Yeah, what would?
That could rapidly undermine how effective piperaquine is for those very prevention strategies. Its usefulness for mass prevention could decrease, maybe quite quickly.
So the tool we're using for broad protection might actually be speeding up the process that weakens the tool itself.
Yeah.
That's a tough situation.
It really is. And it leads to another point the researchers brought up thinking about future drug combinations.
How so?
They suggest that ideally, combination therapies should use drugs that stick around in the body for roughly the same amount of time. Similar pharmacokinetics is the technical term.
Ah, so you don't get that long period with just one drug left over.
Precisely. To avoid that extended window of single drug pressure, which seems to drive the selection for these kinds of tolerance or resistance mechanisms.
That's a smart consideration for future drug development. Okay, but like all studies, this one has limits, right? What should we keep in mind?
Definitely. It's important to remember this was looking back retrospectively at data from one specific study in southern Mali.
So not necessarily true everywhere.
We can't just assume these exact findings apply automatically to other regions with different malaria patterns, different parasite populations, or even different human genetics. We need more research in other places to see if this holds up.
And what about understanding the how? Did the study fully explain how the plasmepsin gene copies provide protection?
Not entirely. It showed a strong link, a correlation between more plasmepsin 3 copies and weaker piperaquine protection, but the exact biological nuts and bolts of how that works, that wasn't fully figured out.
So there could be other factors involved too.
Yes, potentially other genetic changes or complex interactions within the parasite that contribute. More digging is needed to understand the precise mechanisms.
And also, the malaria landscape itself has changed since 2012-2014, hasn't it?
That's a very important point. Back when this trial ran, resistance to the artemisinin part of the combination wasn't a huge issue in Africa.
But it is now. in some places.
Yes, unfortunately, we're seeing the spread of mutations in the Kelch13 gene linked to artemisinin resistance in parts of Africa now. So if the partner drug, piperaquine, is also getting weaker,
Then the whole treatment is much more likely to fail.
exactly, the risk of complete treatment failure could really increase in areas where you have both weakening piperaquine protection and emerging artemisinin resistance. It's a potential double whammy.
Right. So what did the authors themselves, the experts running the study, say about all this?
Well, Pedro, the senior author, really emphasized that their work shows how repeated treatment can lead to parasites developing this drug tolerance. He specifically pointed to the plasmepsin 3 gene duplication as the way parasites manage to reinfect people sooner, which reduces piperaquine's preventative value.
And do they offer advice based on this?
Yes, they strongly recommended, as we touched on, being cautious and having really active surveillance systems in place if piperaquine is going to be used for those large-scale prevention programs.
Like the ones for pregnant women and kids?
Right. Because of that risk of quickly selecting for plasmepsin 3 duplications and potentially losing the drug's effectiveness for those purposes much faster than we'd want.
Makes sense. And are they planning to follow up on this? More research?
They are. They mentioned planning a new clinical trial. This one will directly compare piperaquine with sulfadoxine pyrimethamine. That's another drug used for preventing malaria in pregnancy.
What are they hoping to find out in that new trial?
They want to see if using piperaquine repeatedly in pregnant women does clearly select for parasites with those extra plasmepsin 3 copies, leading to less protection. And maybe crucially, they want to test if alternating between piperaquine and sulfadoxine pyrimethamine could be a strategy to slow down or prevent this selection pressure.
Ah, switching drugs might prevent the parasites from adapting so easily to one.
That's the hope. It seems like a really vital next step to figure out the best, most sustainable way to use these important medicines.
Definitely. So boiling all this down, the study, the implications, the expert views, what's the main takeaway for someone listening?
I think it really forces us to confront this difficult question. How do we keep our essential malaria drugs effective for the long haul? We have to think hard about whether our current strategies, even ones with clear short-term benefits, might be unintentionally speeding up drug resistance.
It's that classic balancing act, isn't it? Immediate protection versus long-term sustainability.
Exactly. Balancing the good piperaquine does now with the risk its long presence poses for future parasite evolution.
So as listeners process this, what should they be thinking about?
Maybe consider how we might need to adapt to malaria control. How can we use these drugs smartly? What's the role for continuous research and, crucially, surveillance watching the parasites on the ground to guide our actions? It really emphasizes this constant chess game between us and the parasite.
Yeah, it's a dynamic battle.
Yeah.
If people want to learn more after hearing this, where could they look?
Well, they could definitely explore the wider topic of antimalarial drug resistance, it's a huge field, or learn more about how different malaria drugs actually work inside the body. And, of course, keeping an eye on the ongoing search for new drugs and new prevention methods is always important. Understanding parasite evolution itself is fascinating, too.
Absolutely. It's a complex challenge that requires ongoing vigilance and innovation. Thanks so much for walking us through this research and its implications.
You're welcome. It really highlights how critical this kind of scientific work is for tackling major global health threats, like malaria.
Indeed. It shows why staying informed about these evolving challenges and the science trying to solve them is so important.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.